Welcome to the set of lectures on Mendelian genetics. We saw that many diseases have a genetic basis. For example, in our country, sickle cell anemia or sickle cell disease, beta thalassemia, Down syndrome, glucose 6-phosphate dehydrogenase deficiency are all occurring genetic diseases, and in other countries such as the US, cystic fibrosis is a widely occurring genetic disorder. So are Huntington's disease and achondroplasia and so on and so forth. These are just examples. Our overall aim was that if we can predict a child's chances to inherit the disorder, the parents can be better informed to handle it in their child. We know quite a bit about the way the information is inherited, or the characteristics are inherited and so on. Whereas for this kind of an aim, the principles of Mendelian genetics, which were developed about a century and a half ago or more than a century ago, are very convenient to use, and that is the reason why we are looking at Mendelian genetics here. I had pointed out I had pointed this video to you. Please take a look at this video if you have not already done so. Slightly longish, but a very nice video on Mendel's experiments and principles. Before we went into Mendel's experiments, we needed to map whatever terminology that we use nowadays in the context of genetics to the equivalent terminology that goes with a Mendelian analysis of inheritance. For example, we had taken cystic fibrosis and we said presence of a chloride transporter. This is rather molecular in nature. It could be somebody being tall and short and so on and so forth. That could also be, and we said that this was a character which is an inheritable, which was a heritable feature, and the presence of the chloride transporter, it could be of two kinds. If the membrane protein for chloride transporter is functional, then it is fine. If the membrane transporter for chloride transporter is not functional, then we get the disease. Therefore, there are two traits, the functionality and non-functionality, and these two traits are variants of this character, and that is the terminology here. In other words, in terms of classic terminology, tall and short, these are the traits of the character height. Then we said that each trait is determined by two alleles on homologous chromosomes, which are the equivalent of genes. For example, this could be determined by capital T, capital T, small t, capital T, or small t t, depending on what is present on these two loci, the locus here, these two loci of the homologous chromosomes. If both capital T's are present, then the membrane protein for chloride transport is functional. If at least one of them is capital T, then it is functional. If both are small, then it is non-functional. This is the mapping between a molecular aspect that we know of nowadays to the olden times, the Mendelian times, when nothing of this was known, and it is really insightful to come up with this kind of a model to explain inheritance. And as you can see, this is a simple approximation. There are various different diseases that do not follow this approximation. We will first look at this approximation, which is very useful, and then I will extend that to show you the differences. I think this is where we stopped last time. So, let us go further with the lecture material of this lecture. We are going to review, or we are going to see some of Mendel's work. Again, let me emphasize the watching of this video. Mendel worked with pea plants to discover the principles of inheritance. In the very first lecture, the introductory lecture, I had mentioned about studying something for the sake of it, studying something because we want to understand something that exists around us. This study was one such kind, and look at where it has led to. Coming back to Mendel's work, Mendel studied 7 characters, each of which had 2 traits. The characters that he studied were flower color, seed shape, pod color, pod shape, stem length, and the flower position. Let us go further to explain this, and these characters, flower color, seed shape, pod color, pod shape, stem length, and flower positions are called phenotypes. For example, the flower color could be either purple or white, both are phenotypes, flower color is a phenotype, and so on. Here we see it in a pictorial form. In the particular case of pea plants, the flower color would either be purple or white, the seed color would either be yellow or green, the seed shape would be round or wrinkled, by round it is actually smooth, round or wrinkled. The pod color could either be green or yellow, the pod shape could be either inflated or constricted, the stem length could be either tall or dwarf, and the flower position could be either axial or at the end terminal. Okay. So, these were the characters that Mendel studied. Most of these characters were rather simple, it just happened by chance that they were simple, and therefore, the basic principles of inheritance could be became amenable to study by Mendel. He could come up with the basic laws of inheritance, most of which was applicable for simple inheritance. Now, let us look at one character, okay, which is flower color. Mendel started his experiments with true breeding plants. What does true breeding mean? It means, in the context of flower color, the same color of flower results in all the offspring in all the generations, okay, which means it is true breeding amongst itself, and therefore, if it is purple, if the flower color is purple, it is purple throughout in all the generations forward. If it is white in all the generations forward. Such plants are called true breeding plants, and Mendel was careful to spend the time to achieve to make sure that they were indeed true breeding plants before he started out his experiments. So, when he started out with true breeding plants, the ones that yielded purple color flowers and the ones that yielded white color flowers, and he crossed them with each other. The pollen and the female part of the plant were made to interact in different ways, and this cross resulted in a purple flower in all the plants of the first generation. This is the parent generation. The first generation had only purple flowers. There was no sign of the white flower at all, and there was nothing in between. It was either purple, rather in this case it was purple or white, and in the F1 generation it was all purple. This was the observation, and he had observed this over a large number of plants, and he did a statistical analysis to find that statistical analysis of course does not mean much here. He did statistical analysis over the entire set of experiments to come up with these various findings. Here it does not make a difference, here everything had only purple flowers. And then what he did was he either self pollinated or cross pollinated the purple flowers amongst themselves. Okay. This crossed with another purple flower of the F1 generation itself. So, when that happened, in the F2 generation something strange happened. 3/4 of the flowers, he had studied about 900 and something plants, here among 929 plants, about 3/4 were purple and 1/4 was white approximately, a very large number of plants. So, the white color which was missed in the first generation made an appearance in the second generation. That is what he found. So, after a lot of experiments with a lot of single characteristics, dual characteristics and so on and so forth, Mendel came up with a model to explain the results, which can be given as follows. Alternative versions of genes or alleles, say capital P for purple and small p for white, determine the variations in inherited characters. This is the first aspect of the model. In other words, the genotype, genes or alleles, determine the phenotype, which is the observed character. The P determines whether it is purple and the small p determines whether it is white. For each character, the organism inherits 2 alleles, one from each parent. If the 2 alleles differ, capital P, small p, instead of both being capital P or both being small p, the dominant allele P determines the phenotype. Okay. For example, if there is a capital P and a small p, the trait of capital P is what would be seen. In other words, this would result in purple flowers. In terms of terminology, if both are the same, either both capitals or both smalls, then they are considered homo-same zygous, okay, they are the same, and whereas if they are different, they are called hetero-different zygous. Okay. Homozygous and heterozygous, these are commonly used terms, it is nice to know. If both are the same, homo, so homozygous; if both are different, hetero, so heterozygous. The 2 alleles for a character separate out or segregate during gamete formation, right. You would have gone through meiosis, you would have studied meiosis, so you know what a gamete is. This is what results during in the cell division that is gone through by the germ cells. Here we are talking about the separation of the 2 alleles, and what Mendel said was that they segregate during gamete formation and are placed in different gametes, so the egg and the sperm cells. Okay. And this is called the law of segregation. The 2 alleles separate, they are placed in separate gametes, and each character, for example, flower color, seed color, seed shape, these are all different characters. Each character is inherited independent of the other characters. Right now we are looking at only one character, but all characters are being inherited simultaneously, that we all know. And what Mendel said by through his studies was that each character is inherited independent of the other characters, and this is called the law of independent assortment. And these two are supposed to be major contributions, the law of segregation and the law of independent assortment. Why this is so will become clear very soon. It lends itself to very easy analysis. Therefore, if we look at the earlier experiment again, the P generation had purple flowers, white flowers. The purple flowers, these are true breeding varieties, and therefore the purple flowers means both are capital P and the white flowers means both are small p. Okay. In other words, there is no heterozygosity here. So the gametes would have both capital P in this case and both small p's in this case, and upon fertilization the genotype is capital P small p, which results in a purple flower because capital P is dominant, right. And the gametes from this would have, half of them would have capital P and half of them would have small p, and therefore in the F2 generation, if you look at all possibilities, this is, let us say, from the sperm of the plant and this is from the eggs of the plant, capital P small p, capital P small p, and the combinations would be both capital P's. Here it will be capital P small p, here it will be capital P, this is the eggs, which is this is P, both are P, capital P small p, and therefore this will be capital P small p and this will be both small p's. Okay. If at least one is a capital P, then it will result in a purple flower because it is dominant, and therefore, you have 3 out of 4 being purple and 1 out of 4 being white. Okay. This kind of an analysis, where you look at all possibilities by using this kind of a square with the gametes from the sperm and gamete from the eggs, is called a Punnett square. Okay. This is heavily used in this kind of analysis. It is rather nice if you are not dealing with very complex situations. 1 or 2 characters, it is quite good to do. It gives you a visual feel for what is happening. Okay. Whereas, if you are comfortable with probabilities, that is a much easier way to do. Okay. We will look at that in a little while. Now, let us look at probabilities. It is, as I said, if you look at it in terms of probabilities, if you are comfortable that way, then it becomes much easier to do the analysis. Let us apply this, or let us look at whatever we did just now, the cross that we did just now, in terms of probabilities. For the cross between true breeding plants that we just considered, in the F1 generation, the probability of getting purple flowers was 1. Okay. All were purple, either resulting from arising from capital P small p. Okay. So, monohybrid, mono is 1 character, hybrid means it is a mixture of 2 allele types, and the probability of getting white flowers is 0. Whereas, in the F2 generation, the probability of getting purple flowers is 3/4, arising from either homozygous condition or heterozygous condition. Capital P homozygous, capital P small p in both these kinds is heterozygous. And the probability of getting white flowers was 1/4, arising from only small p of all the possible combinations. And therefore, the probability of getting a heterozygote in F2, right, is 1/4 plus 1/4, which is half, and so on. You could do various different calculations of this kind in terms of probabilities. You can find the probability of various different things. So, you do not have to go and draw squares, which is somewhat elaborate and so on, if one is comfortable with using probabilities. It becomes much easier when we use probabilities, especially because according to Mendelian principles, the inheritance of characters are independent of each other. Okay, law of independence. Thus, we can analyze probabilities of simultaneous inheritance of multiple characters by considering them as independent events. Okay, it will become clearer here when we consider 2 characters. And now, let us see the way Mendel saw it. For that, let us look at seed color, yellow or green, yellow capital Y, green small y, and seed shape, round capital R, wrinkled small r, being inherited together or being considered together. Okay. Same kind of a cross, true breeding, therefore, capital Y, capital Y, capital Y, capital Y, capital R, 2 characters here, seed color and seed shape, and small y, small r, small r. The gametes in this case would be capital Y, capital R, the gametes in this case would be small y, small r. Upon fertilization, it is going to be completely heterozygous, capital Y, capital R, small r, and then they are going to be independently inherited or independently assorted. Therefore, the seed color would be independent of seed shape, the inheritance of seed color would be independent of the inheritance of seed shape. That is what it means. If we draw a Punnett square here of all the possibilities, you can see, if you count, that 9 out of the 16 possibilities would be yellow and round, both dominant characters. 3 out of 16 would be round and green. 3 out of 16 would be yellow and wrinkled, and 1 out of 16 would be green and wrinkled. Okay. This is how it happens with 2 characters, which we can get an idea by using the Punnett square here. In terms of probabilities, probability for a plant in F2 with homozygous dominant genotype for both characters. What does it mean? Homozygous dominant genotype. Homozygous means both should be the same. Dominant means in our terminology both should be capital, right. So, in other words, probability of capital Y and small capital R. We know that the probability of capital Y is 1/4 and the probability of capital R is 1/4, and therefore, since these are independently inherited, independent assortment, you can multiply these 1/4 that is 1/16. Okay. Independent events you could multiply the probabilities. Now, the probability of a plant in F2 with capital Y, capital R genotype, okay, these two are independent. Therefore, the probability of capital Y and into the probability of capital R half into 1/4, that is 1/8. One more thing, just to illustrate how you could use this, it becomes very easy if you are comfortable with probabilities. The probability of a plant in F2 with homozygous recessive genotype for both characters, homozygous both the same, recessive both small letters in our terminology. In other words, probability of small y, small r. We know that this is 1/4 and this is 1/4, and therefore, the probability in F2 with for a plant in F2 with homozygous recessive genotype would be 1/4 into 1/4, that is 1/16, and so on and so forth. You could do a lot of probability calculations with this approach. You could also bring in 3 characters and so on and so forth, 4 characters and so on, then you can work this out. There is 3 characters here. Let us say that we are interested in the following 3 characters: flower color, seed color and seed shape in the offspring from a cross between capital P, capital Y, capital R, and this is heterozygous in P, whereas homozygous in Y and R. So, this is a trihybrid and this is heterozygous for the flower color, whereas homozygous recessive for the other two. And let us say that we would like to know the fraction of the offspring that exhibit recessive phenotypes for at least 2 of the 3 characters. This is what we are interested in. Okay. If you go through the Punnett Square, it is some amount of work, whereas if you know probability, you can quickly write down this. Okay. 2 of the characters should be recessive. Okay. Therefore, it could be either P recessive, Y recessive or R recessive, exhibit recessive phenotypes for at least 2. Okay. So, taken 2 at a time. So, in this case P and Y are recessive, the probability is 1/4 for P being recessive, half for Y being recessive. You can go back and check that, and that has to be combined with this. If only 2 are going to be recessive, this has to be dominant. Therefore, capital R, small r is one possibility, and that is half. Multiply all these things together because of the law of independent assortment, you get 1/16. This is one of the possibilities in which this offspring that exhibits recessive phenotypes for at least 2 of the 3 characters would arise. The other possibility is P and R being recessive. So, if you work out the probabilities for this combination 1/4, half and half, multiply all together 1/16. Another possibility is Y and R being recessive, that is, and the first one being heterozygous. So, half and half, that turns out to be 1/8. Just to keep the denominator the same, let us write it as 2/16. Here is the other possibility, Y and R being recessive, both homozygous, whereas this one is dominant, homozygous dominant. This is heterozygous dominant, this is homozygous dominant, and 1/4, half, multiply all 1/16, and this is all recessive. It says at least, for at least 2 of the 3 characters, 2 recessive phenotypes. So, all 3 can be recessive. So, that those probabilities are 1/4, half and half for small p, small y and small r, multiply all together 1/16, right. So, in all these ways you could get offspring that exhibit recessive phenotypes for at least 2 of the 3 characters. So, you just have to add all these things to get the total probability. So, probability of at least 2 recessive phenotypes is just addition of all these probabilities that turns out to be 6 by 16 or 3 by 8. So, a lot of probabilities this way are found and they turn out to be useful, as we will see in some ways later on when we discuss diseases. If you look at what has happened in terms of what is happening during meiosis, this is what happens. The P generation, here I think a slightly different example is considered. Yellow round seeds, these are 2 traits being considered together, that is fine, and these are green wrinkled seeds. Yellow round seeds, true breeding, therefore capital Y, capital Y, capital Y, capital Y, capital R, and green and wrinkled would be small y, small y, small r, small r. During meiosis they form gametes. These gametes would only be capital Y, capital R. These gametes would only be small y, small r, and upon fertilization these are the 2 possibilities. Let us look at them one by one. They get mixed up, and by the way they arrange themselves, it would be capital R small r here, capital Y small y here. Please go and take a look at what happens during meiosis, especially during metaphase 1 and anaphase. In the anaphase these start separating out, and metaphase 2 you have these gametes being formed, capital R, capital Y and small r, small y, and therefore, the gametes here would all result in either capital Y, capital R, these 2, or small y, small r. Okay. Alternatively, if the arrangement is like this, small r, capital Y, capital R, small y, then during anaphase they segregate out, resulting in small r, capital Y, capital R, small y, and small r, capital Y, small r, capital Y here, capital R, small y here. Therefore, 1/4, 2 out of 8 is capital R, capital Y, 1/4 is small r, small y, 1/4 is small r, capital Y, and 1/4 is capital R, small y. This is essentially what has resulted from this kind of a segregation. This will become clearer when we look at 2 things together, but this gives you the molecular basis for the Punnett square itself, in this case 2 characters, but you could also look at it in terms of 1 character, nothing wrong. I think this is a right time to close this lecture. Here we saw Mendel's experiments with pea plants, their results, and analysis of those results using simple probabilities and the law of independent assortment that Mendel found. When we move forward, we will take things further towards diseases and see the use of Mendelian genetics to predict diseases. See you in the next lecture.